antimicrobial activities of aldehydes and ketones produced during rapid volatilization of biogenic oils. This document delves into the various aspects of disinfectants, exploring their mechanisms of action and their effectiveness against microorganisms. The study begins by discussing the different types of disinfectants. Then it delves into the mechanisms by which these disinfectants work, highlighting how they interact with cell surfaces, penetrate cells, and target specific sites within the cell to disrupt microbial processes. A significant focus of the research lies in exploring the antimicrobial properties of biogenic oils, specifically fatty acids, esters, and methyls soyate. These natural compounds are presented as potential alternatives to traditional disinfectants, offering advantages in terms of reduced toxicity and environmental impact. The study emphasizes the potential of this biogenic oil as effective and sustainable antimicrobial agents. Furthermore, the research examines the complex nature of biofilms, which are communities of microorganisms encased in a protective matrix. Biofilms are known for their resistance to antibiotics, and other antimicrobial agents, making them a significant challenge in healthcare and other fields. The study investigates the mechanisms that contribute to this resistance, including drug efflux pumps, which actively pump out antimicrobial agents, and diffusion barriers, which restrict the penetration of these agents into the biofilm matrix.
Statement of objectives. Evaluate antimicrobial properties of net compounds. The primary objective was to evaluate the antimicrobial properties of commercially available net compounds, aldehydes, and ketones to validate their potential as disinfectants. Determine headspace equilibrium. The study aimed to determine the headspace equilibrium of the net compounds to understand the relationship between their liquid and vapor phases under antimicrobial activity. Assess antimicrobial property of methyl soyate derived vapors on biofilms. The research sought to evaluate the effectiveness of vapors produced from methyl soyate against biofilms formed by bacterial strains Klebsiella pneumonia and Staphylococcus epidermidis.
Experimental disinfection activity of volatile compounds. The study involved exposing pre-inoculated agar plates to various concentrations of net aldehydes and ketones in a controlled environment. Disinfection activity of oil, vapors, and biofilms. The research investigated the antimicrobial activity vapors generated from methyl biofilms grown on microscope slides.
Exposure to methyl soyate vapors. The bioflame where the exposure of methyl was soyate vapors generated to control temperature for varying duration. Operation of smoke generation. The research utilized a smoke generation to produce a methyl soyate vapors as a specific temperature and flow rate disinfectant vapor generator. Cleanup. The study emphasized the importance of the true cleaning procedures that prevent cross-contamination during the experiment. Determine the headspace equilibrium using CGFID. The research employed gas chromatography, flame ionization detection or CGIFD to quantify the headspace concentration of the aldehydes tested.
Effect of varying concentration of hexanal on bacterial survival. The study found that hexanal exhibited antimicrobial activities against Salmonella typhimurium. but only at high concentration and extended exposure of time. Effect of varying concentration of heptanol on bacterial strains. Heptanol also demonstrated antimicrobial activity against Salmonella typhimurium with a similar concentration and time dependence as hexanol. Effect of varying concentration of pentanol on bacterial strains. Pentanol showed a less pronounced antimicrobial effect against Salmonella typhimurium compared to hexanol and heptanol. Effect of varying concentration of propanol on bacterial strains. Propanol exhibited limited antimicrobial activity against Salmonella typhimurium. Disinfectant activity of mixed aldehyde. The study investigated the synergistic effect of mixing hexanol and heptanol, but no significant increase in antimicrobial activity was observed. The disinfectant activity of aldehydes and ketones mixture. The research explored the antimicrobial activity of a mixture of aldehydes and ketones, but again, no significant synergistic effect was observed at concentration 10 times higher than those found in methyl soyate vapor. Determination of headspace equilibrium. The study used GC-FED to determine the headspace equilibrium of hexanol and heptanol, revealing that the effective concentration for antimicrobial activity were lower than the initial liquid concentration used. The disinfectant activity of methyl soyate on biofilms. The research demonstrated that methyl soyate vapors effectively killed biofilms formed by Staphylococcus epidermidis and Klebsiella pneumonia.
Conclusion: metals with vapor as the effective disinfectants. The study conclude that vapors generated from methyl soyate are effects disinfectant against a wide range of bacteria, including those found in biofilms. Synergistic effects of chemical components that research suggests that the antimicrobial activity of the methyl soyate vapor must mirror the synergistic effects of multiple chemical components rather than the individual activity of any single compound. Further research needed. The study highlights the need for further research to identify all the chemical components in the methyl solid vapors that and the investigate their individual and combined antimicrobial effects.